Adrenaline Signaling Pathway by Audrey and Ava. Epinephrine is a hydrophilic signaling molecule used in the epinephrine signaling pathway. GPCRs are involved in sensory perception, including the senses of taste, smell, and vision. Many hormones such as adrenaline, insulin, and serotonin exert their effects by binding to GPCRs on target cells. This binding triggers intracellular signaling pathways that regulate various psychological processes, including metabolism, blood pressure, and mood. Some advantages of GPCRs are they can respond to a wide variety of ligands, including hormones, neurotransmitters, and odorants. GPCRs also play a central role in transmitting signals from the extracellular environment to the inside of the cell and can regulate a wide range of cellular responses. A single ligand receptor binding event can trigger the activation of multiple downstream signaling molecules, leading to a widespread cellular response. Some disadvantages include GPCR signaling pathways can be quite complex, and it can make it challenging to fully understand and manipulate these pathways for therapeutic purposes. Also, targeting specific GPCRs without affecting others with similar ligand specificity can be a challenge. Lastly, if there is a mutation within a protein in the signal transduction pathway, it will disrupt the whole cellular response. GPCRs work by a ligand binding to the GPCR embedded in the cell membrane. Next, the ligand induces a confrontational change in the receptor, and this activates the GPCR and allows it to interact with other proteins inside the intracellular space. Next, the activated GPCR interacts with the G protein and causes it to exchange GDP for GTP, leading to the activation of the G protein. The activated G protein can then activate various enzymes, which can then produce second messengers, including cyclic AMP, IP3, etc. The second messengers, for example CAMP, will then activate pKa. Then, the previously activated pKa can attract specific target proteins by adding phosphate groups on them. Lastly, the phosphorylation of the specific target proteins can induce a widespread cellular response.
The adrenaline signaling pathway in a liver cell occurs when the adrenal glands release adrenaline when a stimulus is present, and adrenaline binds to the GPCR. The adrenaline molecule activates the receptor, which then activates the G protein. Alpha subunit of the G protein dissociates off. GDP gets switched out, and GTP replaces GDP in the alpha subunit. The alpha part of the G protein continues to diffuse across the membrane before encountering adenylyl cyclase. Second, adenylyl cyclase is now activated. Adenylyl cyclase then converts ATP to CAMP, only has one phosphate group. Continuing the signaling cascade, CAMP travels through the cytoplasm and binds to pKa. pKa is now activated and can phosphorylate certain proteins. pKa will phosphorylate phosphorylize. Phosphorylize is an enzyme that is now activated and can now produce glucose from glycogen. Glucose made from phosphorylize gets secreted out of the cell by membrane-bound transport proteins, glucose transporters. Cellular response. Glucose that gets secreted in the final step of the transduction pathway travels to different tissues and organs through the bloodstream. In many tissues, glucose is taken up by cells and used as an energy source during a stressful situation. Glucose will provide the necessary energy muscle cells need to contract. Intended response. Increase alertness, heart rate increase. A rapid pumping of blood will supply the body with oxygen and nutrients. Dilated airways cause bronchioles in the lungs to expand, allowing for increased oxygen intake. Increased energy. With an increase in glucose, cells have more available energy to use in cellular respiration. How is the adrenaline pathway terminated? As adrenaline concentration decreases, it begins to dissociate from the adrenergic receptors. When the ligand is detached, receptor deactivates. ATP gets switched out and GDP replaces GTP. Alpha subunit of the G protein then reattaches to the two subunits. Without the activated G protein, adenylyl cyclase is inactive and stops making CAMP, which inactivates pKa. Phosphatase enzymes dephosphorylate target proteins that were phosphorylated by pKa during the signaling cascade. Cellular responses initiated by the adrenaline signals start to reverse. For example, the heart rate, which was increased, returns to a normal resting rate.